Perfect. All right, good morning, everyone, and welcome to the first ever MIHRA Clinical Trials Site Community Forum. I'm very happy to welcome you today and welcome all of our fantastic speakers and moderators. Today's meeting, it's going to be jam-packed with a lot of information, several videos, and multiple discussions. So I just want to go through a couple kind of ground rules to make sure that everyone gets the most out of the meeting today. So if you are having any technical difficulties, video or audio not working, if you're having trouble with the chat feature, please be sure to log out of the Zoom account and log back in. If the issue still persists, you may want to log into your Zoom account directly and change the settings from within. I'd also like to introduce our forum co-chairs, Julie Pack and Pedro Machado. I'd also like to give a special acknowledgement to Antonia Valenzuela, who is also a co-chair but was unable to join us today. With that, I'd pass it to our co-facilitators.
Great, thanks so much, Angel. Welcome everybody, and I don't know if Pedro's here, but yes, I'm one of the co-chairs. And I am an adult rheumatologist at Johns Hopkins, and we're very excited to get this first community forum up and going. Pedro, I'll let you introduce yourself.
Hi, everyone. So my name is Pedro Machado. I'm a rheumatologist, adult rheumatologist based at University College London. And thank you all for joining, and welcome to this forum.
Great. So we just want to give you a little bit of background about MIHRA and the CTSN, or the Clinical Trials Network. It was really incepted by the community members in 2017. And we really believe that all the revenue generated through this sharing site and the data with industry, for example, like expedition of site outreach and feasibility mapping, would all be directly invested into the research community through project grants and fellowships and even career development. And so this very concept led to the formal discussion transitioning that important myositis research community into a viable funding infrastructure. And ultimately, I believe that is why we became to be what MIHRA is, and this concept of CTSN became to be. And we really, truly believe, I know I've been saying we really believe, maybe this is my California upbringing, but we truly believe that there's this, our MIHRA CTSN is built for inclusion and cultivation and really support of the up and rising, myositis investigators and trialists, but also senior ones where we can really learn from them and strengthen our operations in the future. So today's forum is really specifically focusing on CAR T therapies, not bispecifics or the BiTEs that we are also hearing so much about. And I think this is a very exciting area because, people have questioned us, is this going to be the cure-all and be-all? But I think we need to learn more about it. And we believe that even within the myositis community, understanding the different concerns and the questions and the struggles we may undergo through within our own institutions, we want to have an open discussion about this. So we hope you enjoy our first community forum. And just a reminder, this is not an accredited educational event, but we just want to bring the community together. And please don't forget to write down any questions in the chat, and both Pedro and I can, field those questions as moderators and co-chairs. Pedro, I don't know if you want to say anything else. I'm sorry if I...
No, that's perfect. And I guess so. As the first step for this forum, we'll begin with a video of perspectives from our colleagues. And I think it's now time to watch the video. Oh, Leslie has a
I just
comment.
would like to say it's not so much questions in the comment box, in the chat box. It's everybody's thoughts, reactions, responses, concerns, hopes, excitement. Continually feel to use the chat to respond and express yourself.
Yes, yes.
So, Angel, would we please have the recorded video about perspectives from colleagues? Thank you.
name is Ingrid de Groot and I have dermatomyositis. Besides being a patient, I'm also a patient research partner and a patient advocate. Recently, we, the people with myositis, are being bombarded by snippets of publications, by promising news flashes and screaming headlines regarding CAR T-cell therapy. Is that a good thing? Yes and no. Yes, because many people with myositis are desperately waiting for an effective and a long-lasting treatment, and this CAR T-cell thing may be just that. We all know that hope springs eternal, but that makes us also very vulnerable. And that is why this trend is not just all a good thing, because most people do not exactly know what we are talking about here. And in patient groups and on social media and in in-person meetings, the topic is really hot, which is great because we really need to share those opinions, share those hopes and address those concerns. And of these, we hear many of them overlapping as well. Because what are we exactly talking about here? It might be working in cancer, but myositis is not cancer, is it? What are the safety risks? What are the risks in the long run? We realize no one can tell at the moment because most patients with myositis have only recently been treated. Are there side effects? What exactly is involved here? Are we talking just infusion therapy here? And it is very, very hard to find lay information, and we do not even know if this information is reliable or not. And that is a worry in itself, because we all want the same thing here, and that is offering patients with myositis the best of care and maybe even a cure. And treating patients requires mutual trust and shared decision-making. But as things are now, many patients feel not sufficiently equipped and confident enough to make such a crucial and significant decision about this potentially life-changing treatment.
Thanks very much for the opportunity to record this video. We have increasing interest in this area, and we've been exploring a number of cellular therapy trials in the context of autoimmune diseases at our institution, one of which is set to commence soon. Our institution is co-located with the Peter MacCallum Cancer Centre, which actually hosts Australia's largest cancer immunotherapy research program. And so we have access to significant resources and subspecialty expertise in this regard. Nonetheless, we face a number of challenges. Our primary challenge is that our rheumatology unit is relatively small and we lack infrastructure required to conduct clinical trials independently. And thus we are required to run our trials under other units where staff may have limited familiarity with our specific diseases. With regards to the trial protocols, we have concerns about the lymphodepletion regimens typically required and the potential associated toxicity. And another specific challenge that has been identified is the need to wean DMARDs and corticosteroids for a period of time prior to lymphodepletion, which increases the risk of flare during that time. There's some apprehension about the potential toxicities of cellular therapies, such as cytokine release syndrome, neurotoxicity, but as well as the potential unknown long-term effects, such as secondary malignancy. And these risks may be considered less acceptable in the context of rheumatic disease compared to malignant disease, for instance. So as a unit, we're inclined to explore cellular therapy trials that have been designed to be less toxic. Another issue that we have faced is the ever-increasing number of trials in this space, and we're frequently approached by groups offering CAR-NK or CAR-T cell trials, and navigating the relative merits of these options is complex. Thank you.
Hello, everyone. I would like to share my hopes and concerns for CAR-T therapy. According to previous reports, patients with anti-synthetase antibodies who had poor responses to multiple drugs might be favorable candidates for CAR-T therapy. In addition, anti-MDF5 positive Japanese patients frequently develop severe lung disease, leading to devastating outcomes. I hope CAR-T therapy will overcome those intractable diseases. In Japan, the feasibility of CAR-T is limited to specialized institutions. and is currently only approved for the treatment of hematological disorders such as B-cell leukemia and lymphoma. In addition, the medical cost is extremely high. In the future, we need to develop a feasible infrastructure, including specialized doctors, budgets, and dedicated equipment for autoimmune diseases. As a new treatment, we still need more data regarding safety and long-term outcomes of CAR-T. We need to share information on CAR-T with collaborators across the world and identify patients who are suitable for CAR-T therapy. Thank you.
Few problems to solve initially. First, establishing a clinical trial relationship with the hematology and the bone marrow transplant unit in our university. And finally, they allowed me to choose one trial, and we were approached by many, and I was confused. Should I go with an autologous versus allogeneic CAR-T product? Should I go with a traditional CAR-T or a CAR-NK? Or some of the newer products. Also, the option of off-the-shelf products, which does not need any ablative chemotherapy before the CAR-T. So many choices which are confusing. Then I was also not aware of the basket trials, which has scleroderma, myositis, and vasculitis together. By the time I became aware of that, my scleroderma colleagues already went with another trial. I wish we had chosen a basket trial so that we could all collaborate and use the resources more wisely.
Hi everyone, this is Dr. Kunal Chanwar. I'm a clinical immunologist and rheumatologist from India, and I'll share my point of view regarding the challenges CAR T-cell therapy trials might face with myositis in a country like India. The first and foremost is regarding the infrastructure and logistics for these trials. Limited specialized facilities are available in this country, and even regarding the technical expertise that is needed for CAR T-cell therapy, there are limited experts that are there. Regarding specialized equipments, cold chain maintenance, interdepartment coordination, and follow-up planning is still not that easy in our country. There are financial barriers and access and equity issues. Since we know these diseases affect the disadvantaged populations the most, there are excess concerns that we might not be targeting the patients who actually need CAR T-cell therapy the most, and there are concerns regarding high base therapy costs, hidden costs, and out-of-pocket expenses that are there with this therapy. There are regulatory issues since the current CAR T-cell trials are designed for cancers, and the regulatory framework is again for cancers, to ensure that we have a regulatory framework in place for autoimmune rheumatic diseases is essential. There are perception barriers. These include that this is a therapy predominantly directed to cancers, and if we use it for autoimmune diseases, what implications it might bring about. The concerns regarding post-CAR T-cell therapy, CAR T-cell leukemia is again an issue. And to ensure that we have effective therapies in the long term, a lot of planning and interventions from all stakeholders is needed. Hopefully, we'll have good, effective CAR T-cell therapy trials in this country soon. Thank you.
To live a normal life with myositis over the last 20 years and was recently approached to take part in a CAR-T trial. While it's very tempting to have the possibility of never having to have a regular infusion of IVIG or rituximab again or any other kind of medications and to return to a more normal life, I declined for many reasons. The main concern is that I don't think we really know how this works in patients like me. Will it really work in the long term for my specific type of myositis? Will I cope with the preconditioning therapy, or does this already reset my immune system? will my immune system recover without any long-lasting effects? The known risks and management of CRS and ICANS also terrifies me, despite the medical knowledge that I have. And I know that the cases so far have been very positive, reflecting remission up to a few years and low-grade AEs, but I still feel that we are very much taking a gamble here with the patients that we are trialing this on. Such concerns may not be fully understood or fully explained to a patient who might be very weak and vulnerable at the time that they are approached to take part in such a trial. And I do suspect that sometimes desperation or even sometimes misconstrued promises of a cure may drive some patients to agree to put themselves at unnecessary risk. While I'm very happy to see such strong interest in the industry and the community to find a way to cure myositis, on one hand, I know we do need to study this, but I do fear that the recent high-speed acceleration in trials is really pushing all of us in the community to learn how to fly before we even learn how to crawl and walk. So I think we do need to work together as a community to really address all these questions and difficulties before we start to test things on patients.
Hello, welcome. So glad everybody is here, and just wanted to thank everybody for contributing to the surveys, the open-ended surveys for each investigators and patients and supporters. This is an extremely preliminary analysis, not formal by any means, but just combing through the data and picking up repetitive concepts. We had 58 investigators answering the investigator-specific survey and 24 patients and supporters replying to that open-ended survey, which was a free form. Both of them were free form narratives, so anything could be put in these fields. A wide variety, a wide spectrum of nationalities, geographical spread. You could see here the spread is very, it's skewed to rheumatology, with others what represented. But one thing I would say is that this particular methodology, we're looking at each discrete concept as being important. So there's no frequency weighting on these results at this stage of the consensus building. A strong representation by adult, but very strong representation by the pediatric population, who care for pediatric population. In terms of patients, this is the spread of representation. Inclusion body myositis as a single disease was strongly represented. However, if we look at the inflammatory myositis diseases, if we look at them together, we could see that they were parallel or exceeded that of inclusion body myositis. But again, we're at the very beginning, a very open-ended stage of a methodology to understand community experience and their concerns and suggestions. So there's no frequency weighting on these results at all. So let's look at first what patients shared with us. There's a need for better information, a lot of optimistic adverts. Some patients are desperate for good news, but there's need for reliable information on this topic. The word bombed was used with information on the success, but when delving deeper, studies and small groups were where this information was coming from and what was really known about long-term effects and what exactly is CAR-T and what does it do. Patients expressed serially a detailed desire for detailed understanding. Is it a cure or is it a treatment like rituximab and IVIG? There seems to be not enough data on this. There was the concern. It was discussed that there's a desire to know the story of CAR therapies in cancer, the risks, efficacy, and then how this relates possibly differently or the same in connective tissue diseases and myositis. So in terms of side effects, enough known about it to consider it safe, short-term, long-term side effects, very similar to the concerns that investigators had. Remission, how long? Is it painful? Can it affect organs? And it was voiced repeatedly about the ultimate costs, financial costs to patients. And there was several voicing of the toll, the financial toll on national financial costs and insurance programs, and advice that patients wanted to give to when discussing CAR-T. Clearly, you could see in the bold lettering. These are common sense, but there's enough studies actually to show that we don't do these things well. Respect, empathy, kindness, patience. The more information, the better. Be honest. So there's a fear. It sounds like that they're being bombarded, and we as investigators are being bombarded and being colored by this being dazzled, over-dazzled. Adopt a listening attitude, speak to a patient with respect, but at a level of content sharing for a 6-year-old to understand. Very detailed components of procedures were voiced as being absolutely needed. Why is it potentially better than conventional therapies? And how is efficacy and side effects? What does this mean in respect to the type of disorder a patient has? And what level of functional improvement might be achieved by this therapy? Patients should repeat information in their own words to ensure understanding, have it on paper so patient and loved ones have time to read it, and make sure that physicians are available. I'm going to go through these very quickly, but there was just tomes and tomes of input from investigators, and a lot of it I'm sure is going to come up in the upcoming talk. But generally, there's a pressure from the industry sector, but not enough really time to understand. It was expressed investigators and patients are caught in a whirlwind created by an industry competition, and the baseline data collection is uncertain that it's high enough quality, and we're banking on low trial safety data, but asking a lot of we're asking for high volume enrollment. Sites are being pressured to enroll that have limited or no experience, and that those are sites themselves saying that's us. We have little experience and we're being pressured. How is this impacting enrollment such that there will be sufficient power that after making all of this investment that we're really going to say anything? There's concern about publication bias right now because we're so optimistic and dazzled, and the risk-benefit ratio is extremely hard to evaluate, especially in patients that have not even experienced other treatments. There's a strong concern about sidelining, good word, effective, less expensive, more easily administered, possibly safer therapies, and it's hard to recruit patients for other promising research. And there's concern, again, echoing patient pool getting diluted between multiple different companies and products. Is the end data really going to tell us much? Especially because these are different molecules, logistical impediments. A lot of this surrounds about labor division, budget division, collusion, collaboration with our oncology colleagues, realistic budget projection, because there's such an immense amount of setup time, administrative time, investigative time that does not seem to be covered in the actual costs. And in the end, are the costs of the trial actually going to be sufficiently covered? And a big issue is hospital administration with 10 to 14 days specialized hospital stay, for example, in a bone marrow unit, and that limits access for patients, displaying patients with oncological diseases that have a known calculated benefit based on a well-defined oncological profile. The safety and risk concerns are the common ones that we've seen that I'm sure are going to be voiced today. Again, in CTD, higher propensity for infection, variable malignancies, and induction of autoimmune, alloimmune responses like we see with checkpoint inhibitors, and the poor characterization of antigens. Could muscle inadvertently become a targeted tissue? The very important one to highlight here is there's a lot of concern around patient vulnerability during the interval washout period, and the waiting on manufacturing of cells. Patient could become unstable, and particularly in myositis, who are prone to opportunistic infections, higher risk for those. There's a concern again for exacerbation of muscle damage mediated by CD18 cytotoxic T lymphocytes and secondary malignancies in susceptible hosts, as we know. Exclusion criteria concerns are extensive, and defining what is active disease, and concern for several populations within the myositis group as being well represented, and the disease heterogeneity being an issue with being able to build inclusion criteria and response, and ultimately repeatedly echoed were that these patients have other therapeutic options. Have we really exhausted them? And who are the patients that really need this when we don't have well-established treatment guidance that's more uniform? And we lack a biogenetic profile predictability that's available for oncology patients for selection. So there seems a voicing that a lot of work needs to be done. Thank you very much, and I'm really looking forward to these talks and discussions.
Okay, great. Well, that was wonderful. And we are going to now move to the second part of discussing the clinical trials landscape. We're going to have a wonderful discussion and speaker. So I'm just going to introduce our moderators, Dr. Jisna Paul, and also Karen Chang, that you've already heard from.
Hello, welcome to the next session. I'm Jisna Paul, an adult rheumatologist from Ohio State University. The next session, I would like to welcome Pedro Machado, who's Professor of Rheumatology and Neuromuscular Disease from the University College of London. He'll be giving us an overview of CAR-T therapy and trial implications.
Thank you. Thank you very much. And the testimonies that we've had so far, they've been a fantastic introduction to this forum, and I'll just try to summarize some of those thoughts and also give an overview about CAR-T cell therapy as an introduction to the next section. So I'll share my slides, and you should be now seeing my slides in full screen. Immune-mediated muscle diseases, so the idiopathic inflammatory myopathies, they're truly systemic diseases with a lot of potential end organ damage, not only at the skeletal muscle level, but also at the level of the skin, the joints, the heart, the blood vessels, the lungs, and the GI tract. So there's a lot of potential damage and potential life-threatening complications, a lot of potential for disability and loss of quality of life. And although we have a good range of drugs that we currently use to treat our patients, we also know that these drugs are not always effective, and they do not work in a significant proportion of patients who sometimes partial respond to these medications or don't respond at all. And therefore, there's a need for additional therapies in the idiopathic inflammatory myopathy spectrum. And for example, in IBM, there's still no effective drug treatment for that patient population. And over the last few years, we've really witnessed a huge expansion in terms of clinical trials in myositis. Even before CAR-T cell therapy trials started, we were already being bombarded with multiple other clinical trials, and these included pathways such as targeting type 1 interferon, for example, with JAK inhibitors, toll-like receptor inhibitors, among others, drugs that target co-stimulation of T cells, complement inhibitors, and other types of immunomodulation, such as FcRn inhibitors. And obviously, IVIG was already being used, but there's also several ongoing trials with IVIG. And now, we're already using anti-CD20 antibodies, but then there is the potential for a new target, and that target was CD19. And why CD19? Well, because by targeting CD19, that will allow targeting a wide spectrum of B cells and plasmablasts. And that could potentially lead to better outcomes and a wider depletion of B cells. And that's how CAR-T cell therapy came into play. And these are essentially genetically modified T cells, and they're modified using a viral vector. So the viral vector is used to introduce a new antigen receptor on the T cells, and that's why it's called a chimeric antigen receptor. And then these T cells, they gain the ability to identify a new target. Now, if we think about oncology and malignancies, their target is CD19 because, you know, around 95% of the B-cell malignancies, they express CD19. But we can then translate that to autoimmune diseases where the tumor are the autoreactive B cells. So that's the principle. Let's look at the autoreactive B cells are the tumor, you know, the B cells producing the antibodies. And that's why also it only makes sense to use this type of approach in diseases that are antibody mediated, where there's a clear role for the antibodies in the pathogenesis of the condition. So if you look at the bottom figure, we already have antibodies against molecules expressed on B cells, such as CD20, and we've now developed new ways of therapeutic B cell depletion. There's another potential therapy that is being used, although that's not the topic of this forum, which is bispecific T cell engagers or BiTEs. And so these are a class of bispecific antibodies, and they can, for example, target CD19 on B cells and CD3 on T cells, and then they bring both these cells together, enabling T cell directed cytotoxic killing of the B cells. However, with CAR expressing T cells, these are designed to target surface molecules on B cells. And CD19 is the most common one, but not the only one. And this makes them as capable of effectively depleting B cells. There's been a lot has happened over the recent months, years, in terms of how these CAR T cells are generated. In general, there's a intracellular domain and an extracellular domain. So the extracellular domain has the antigen binding domain, so that recognizes the target, let's say, and then that's linked to an intracellular signal domain by this INCH domain. And then there have been variations about how this intracellular signaling domain has been constructed, but an important point is that at a certain point in time, it started including a co-stimulatory domain that has increased the efficacy of this type of therapy, and then multiple approaches have been tried by different academics and also by pharmaceutical companies. And in terms of the spectrum of CAR-based treatment approaches, this can vary in terms of the transduction method, so that's point A, the treatment procedure type, so that's point B, the cell resources of the product, point C, and then there's new methods also for the target cell killing. Now, regarding the various transduction methods that are used to introduce the genetic information of the CAR into activated cells, these have been mainly viral vectors. But there are some new approaches that are being developed, like lipid nanoparticles, which can deliver mRNA to the T cells. And also, these lipid nanoparticles can be used to deliver gene edited by CRISP. So these are the potential approaches for transduction methods, although obviously the viral approach is the most common one and the one that is most commonly used currently, often with the lentivirus, although retrovirus has also been used by some companies. Now, in terms of the type of procedure, the one that is more often used and the one we know more about is the autologous approach, which is based on the cells of the individual that are extracted from the individual. That's the leukapheresis process. But there's other process being developed, namely the use of allogeneic CAR T cell therapy, where T cells from healthy donors of cord blood or inducible pluripotent stem cells are used. And these are often called off-the-shelf cells because they could potentially be readily available for immediate use. And more recently, there's also some approaches to try to do this in vivo. But I think today we'll focus on the autologous approach, which is the one that we know more about and the one that is more commonly used. And although CAR T cells are the most commonly used CAR-based strategies, it's important also to bear in mind that other cell sources are also under investigation, including gamma T cells and natural killer cells, as shown here at and in the left bottom corner of the figure. Then there are several new methods, and these are all investigational. I will not spend a lot of time on those, but there's a lot of research being done, and these are often called next generation CARs, including armored CARs, and you've got a few names here. But essentially, where the CAR T cell approach is often coupled with a second approach, like targeting certain cytokines or other types of cells, and they've been given various names depending on the approach. Okay, see, this slide illustrates the cell therapy landscape for autoimmune disease, and it's just impressive to see how the field has evolved. And this is not an exhaustive list. There are probably more, and this is probably already outdated. But the indications now include not only conditions like lupus, systemic sclerosis, idiopathic inflammatory myopathy, Sjogren, but in many other specialties, namely neurology with multiple sclerosis, myasthenia gravis, autoimmune encephalitis, etc., etc. So the field is expanding enormously. Obviously, these are mainly phase 1 and phase 2 studies or preclinical studies, but eventually we'll probably see also some late phase studies here. This is the limited list of clinical trials in idiopathic inflammatory myopathies, CAR T cell trials. And although there are significant and important difference between all of them, I guess if we had to say what's the common denominator in terms of the inclusion criteria and the population being targeted, these are antibody positive patients, which is kind of obviously given the mechanism of action, with severe organ manifestations, and this could be muscle, skin, lung, or a combination of all of them, and disease that has been refractory to multiple treatments. And again, the number of treatments vary depending on the clinical trial. And I think it's important to acknowledge that this story probably started here with the group from Germany, and they recently published their case series of 15 patients that they've treated with CAR T cell therapy, including 3 patients with myositis. And again, in this study, and this is an academic study, they included patients with treatment of refractory disease that had a severe and progressive disease course, namely in terms of the myositis patients, they all had that active myositis and also evidence of interstitial lung disease, so both muscle and lung involvement, and they had been resistant to multiple treatments. And these are the 3 patients that they treated in Erlangen in Germany, and you can see here that there was lung and muscle involvement, and that they had been treated with multiple drugs. All these patients had been treated with at least 4 or 5 drugs, and some of them even like 6 or 7 drugs, so resistant to multiple treatments. And you've heard about the outcomes. I won't go into a lot of detail about this. The outcomes that were reported were very good, with improvement of strength, function, and reduction of CK levels. So in summary, very promising data, but also significant concerns about the future, which patients should be eligible for this type of treatment in terms of medium long-term outcomes. What is the safety profile of this therapeutic approach? Having said that, we do have important information from hematological malignancies. What we don't know is in what extent this evidence can be translated to autoimmune diseases, because obviously these are very, very, very different conditions. But it's important to bear in mind that more than 34,000 patients have been treated worldwide with hematological malignancies. And for example, in terms of T-cell transformation, only rare cases have been observed among these 34,000 patients. So there's a lot of uncertainty, and that's exactly the reason why this forum was developed. And a few things to consider, and many of these things have been already mentioned today, is choosing the right disease for this type of treatment, selecting the right patient. And to be honest, it's likely that the right patient will vary depending on the development stage of the drug. I think the patients that we will be treating in 2-3 years' time will be probably quite different from the patients that we will be treating now. The choice of target, we know, is it's CD19, it's the B cell activation receptor. Is it what's the role for lymphodepletion? What's the best treatment strategy for lymphodepletion? How should we monitor these patients following CAR T-cell therapy? Or what should we do in terms of concomitant treatments before, during, and after CAR T-cell therapy? How should we manage flares and relapse after CAR T-cell therapy? Should there be a role for another CAR T-cell infusion, if that's the case, for example? And of course, managing toxicities, it's another area of concern. Although from what we know so far in terms of toxicity or short-term toxicity, the safety profile seems to be better compared to hematological malignancies. So a few of the items that I'm sure will be extensively discussed today. And thank you.
Thank you very much, Pedro. Hi, everyone. My name is Karen Cheng. I'm a patient living with myositis. Next up, we have Susan Shenoy, who is a pediatric rheumatologist and clinical director at the Seattle Children's Hospital, and also an associate professor at the University of Washington, as well as Vicki Worth. She's a professor of dermatology from University of Pennsylvania, and both of them will provide their insights on the relevance of CAR-T in children, especially also in children with skin-predominant diseases. Vicki and Susan.
I'm Susan Shenoy. I'm a professor in pediatric rheumatology at Seattle Children's Hospital and Research Center, and I also help run the Seattle CureJM Center of Excellence. I'd like to thank the organizing committee for inviting me to record this video on my perspectives in CAR T-cell for juvenile myositis. Juvenile myositis is a rare orphan disease, and pediatric patients tend to present with more severe vasculopathy, including skin ulcers, as well as they have more severe calcinosis. As many of us know, there currently are no FDA-approved treatments for the management of juvenile myositis, and the pediatric rheumatology community is facing a severe workforce shortage. This leads to delays in access and care for our patients and families with juvenile myositis, which puts an undue burden on them. We as a community in pediatrics are extremely excited about the possibility of CAR T cells for juvenile myositis. There are a few clinical trials that are enrolling pediatric patients for CAR T-cell therapy, and there is actually one published case report for autologous CD19 CAR T-cell therapy in juvenile myositis. This report is a singular case report presented by the Italian group that was recently published. And I'd like to talk about this patient. This was a 12-year-old child with refractory juvenile dermatomyositis. He had ulcerative skin disease and severe myositis, as well as extensive calcinosis. He had failed therapy to chronic long-term steroids, including oral and IV steroids. He had complications of steroid therapy, including a compression fracture. He had failed prior MMF, hydroxychloroquine, as well as IVIG. He'd received 2 rounds of pheresis and 2 courses of rituximab, in addition to 6 months of cyclophosphamide. Autologous CD19 CAR T-cell was given as indicated by the vertical dashed lines here. And as you can see from the before and after pictures, after he received this therapy, he had a dramatic clinical response with resolution of his skin as well as muscle features and significantly improved calcinosis. Several scores, including his physician global assessment, his CMAS, CAT scores, and the interferon scores all improved. As is known with these therapies, this patient experienced mild cytokine release syndrome, transient anemia, and neutropenia. However, encouragingly, he is off all medications at 6 months. The authors monitored for B-cell reappearance, which they noted occurred at about 8 weeks. However, despite appearance of B-cells, this patient's clinical remission persisted, which tells us that CD19 CAR T-cells are able to reset that B-cell compartment and eliminate potential autoreactive pathogenic B-cell clones. So this is really encouraging. Even though it's a singular case report in the pediatric field, it's very encouraging and promising. However, as we move forward, there are a few thoughts that we should consider for our pediatric population. We need to be inclusive and mindful of including and enrolling pediatric patients in CAR T-cell therapies systematically worldwide. We as a group need to identify who is the right patient with juvenile myositis to receive CAR T-cell therapy, and when is the right time to give CAR T-cell therapy to our pediatric patients. And last but not least, when we're giving these therapies to pediatric patients in their teens, we really need to be mindful and thoughtful about monitoring for long-term outcomes that are seen with some of these therapies and be vigilant about learning from what is known through the adult studies. That being said, we at Seattle are very fortunate to have a current trial that is enrolling pediatric patients with lupus for CAR T-cell therapy. And we're hopeful that in the future, we will be able to extend these therapies to other indications in pediatric rheumatology. I'm sorry to miss the conference, but I will wait and watch the recording. Thank you again for inviting me to come share my thoughts.
So I wanted to briefly share perspective, mostly a dermatology perspective with autoimmune disease related to CAR T cells, bispecifics, and siRNA. I mean, there are many ways to modulate the immune system, and there are many more that are under development. And there are different safety issues with all of them. This is looking at CAR T cells, and as we know, there are many different constructs that are now available and being tested in autoimmune disease. And so in terms of the unmet medical needs, in terms of B cell depletion, essentially diseases that need deeper depletion of tissue B cells are a really great target. Diseases where there is a long-lived B cell making antibodies, such as targeting CD38 or BCMA, and diseases where rituximab helps, where essentially the short-lived plasma cells are making the antibodies that are pathogenic. I've listed some of these here, and some of them I put question marks because we have no evidence at all, but we know that rituximab can work for a number of these different conditions. So this is an example of pemphigus vulgaris, and you can see widespread lesions. You can see the flaccid bully and oral involvement, and also mucous membrane pemphigoid, which can cause blindness. And so there's a lot of unmet medical need, and there's some diseases where rituximab doesn't help that much. And I will say, for instance, that skin predominant dermato is one of them. Cutaneous lupus may be a second one. And it's not so clear with monoclonal gammopathies that can lead to things like scleromyxedema or scleroedema. So it seems clear that targeting CD20 can help remove lots of B cells. And again, in the case of something like pemphigus, we know the antibodies are being made by these CD20 positive cells. And we know that now in the era of CAR Ts, we can really be targeting very specifically antigens that will remove different types of these cells. So again, pemphigus works with rituximab, it gets better. But relapses are frequent as memory B cells escape depletion. There's deeper depletion, which may lead to prolonged remissions. And depletion of tissue-resident B cells is a greater challenge than in the periphery. If it's long-standing, maybe longer-lived plasma cells that aren't depleted with anti-CD20. So you have to start thinking about other ways to target the cells that are making the antibodies. And in terms of autoimmune bullous diseases that are not being looked at, there's lots of them. We're not that great at treating many of them. And so in terms of dermatology, I think, basically there are some diseases that would benefit from this approach. I will say that the bispecifics are interesting, although they've got their own set of issues in terms of side effects, but you may have the advantage of off-the-shelf lower cost treatments in the end with no need for lymphodepletion. And I think that will be really helpful in being able to target a broader population. But they're still going to require monitoring for CRS and ICANS, cytopenias, and infections. So we still have issues here in terms of the side effects. And it's not clear that therapies are under development won't meet much of the need. And by that, I mean non-CAR T cells. We have a lot of therapies in the pipeline that are likely going to make a big difference for many patients and potentially be easier to use or cheaper or accomplish what we want to therapeutically. So the safety profile will need to be established in life-threatening diseases, initially at least, and then we can potentially be looking at other more non-life-threatening or less severe disease. And then the risk-benefit ratio will be different for most derm disease relative to other diseases that are perhaps not skin-related. And right now with the conditioning regimen, including cyclophosphamide, fludarabine, if that changes, I think it'll be easier to think about this more in dermatology population. And severe patients may clearly benefit, and in the future may be cost-effective alternatives. We get more off-the-shelf CAR T cells and bispecifics, which combine T cells and B cells simultaneously in direct killing. So I want to really just conclude in saying that there's certainly a lot of need. Certainly autoimmune blistering diseases is one of them. Connective tissue disease, such as dermato, is another potentially. Technology is rapidly evolving, and as safety and ease of delivery develop, we'll likely have applications in autoimmune skin diseases where B cells are important. And so I want to thank you for listening and happy to discuss further.
Thank you. Thank you, Pedro, Susan, and Vicky. At this time, we open the floor for open discussion, and please raise your hand if you have any questions.
Hi, my name is Reza Mears. I'm a rheumatologist in Hamilton in Canada. I think, obviously CAR T cells are very exciting. That's why we're all here. One thing that I think hasn't been discussed as much is what about just targeting, maybe it's not cellular therapy that we need, maybe it is, but what if it's just the target? I haven't seen any trials for CD19. That would be cheaper. There's an off switch. You can stop giving the biologic. There's no chance of this cytokine storm. If someone gets a severe infection, you can stop it. There's no chance of this CAR T-induced T-cell leukemia that you guys have probably read about in the New England in the past few months. So I wonder if anyone is familiar with this.
Reza, do you mean CD19 B-cell therapy, like in ebolizumab or...
Yeah, I've not seen that done
Okay.
in our diseases.
Yeah, I've not seen that as well. Good point.
I agree. I think it's approved for NMO, is it correct? Or one of those diseases. I think there's been some talk of, it's an excellent point, of looking in that space, applying it. I just don't think anyone has done it. I completely agree. So
Yeah, I reached out to a pharmaceutical to propose an investigator-initiated trial about
yeah,
this. And they're like, oh, we're looking into this. And that was the last I heard.
gotta push harder. Okay.
Yeah, exactly. I should.
Yeah, I think it's an excellent point. There's potentially CAR T cell therapy could still have an additional advantage, which is by reaching tissues and areas of the body that the antibodies cannot reach, which has also been a concern with the anti-CD20 therapies. So, for example, with CAR T cell therapies, they can cross the blood-brain barrier, for example, and they could reach some areas of lymphoid tissue that is not reached by monoclonal antibodies, essentially. So that's a potential theoretical advantage of CAR T cell therapy. The other thing is that makes CAR T cell therapy more attractive is this possibility of a reset of the immune system, right? So the possibility of a curative treatment as opposed to another treatment that needs to be given chronically and potentially forever. So yeah, so I think it's a very interesting approach, but and we should probably explore that. But there's still advantages or theoretical advantages from CAR T cell therapy that do not exist with an antibody therapy, and I think these are the main two: the possibility of reaching hidden cells in deeper tissues, and the possibility of a reset of the immune system and a curative treatment that you'll not be able to achieve with an antibody.
I mean, I'll say that in pemphigus, we do sometimes get cures, basically because B cells are reset with rituximab. And in lupus, anti-CD38 has been used and then published. So targeting these different cells types have been done with the antibodies. I do think there's potential for potentially more patients having a longer cure. But I think even in the lupus world, there have been some mild relapses after CAR Ts. And so we have to learn more, to see how this is going to work.
I agree. I think there is a relapse. I think there's a report, and even in myositis, and we don't have the long-term follow-up. I think this is the problem. So I'm kind of just waiting to see what's going to happen too. What is the long-term follow-up, like 14 months, 24 months, or I don't know, for myositis at least. I don't know about the other diseases. Yeah. Definitely there's been relapse with lupus patients, correct? If I remember, there's some reports of that.
Yeah, there have. I mean, I think it's been mild relapses and the patients have been really severe. And so they've gotten a good quality of life in between, but you would hope that a therapy like this would be, truly curative and not, so we need to learn how often it's not.
I think Karen put a, oh, that's great.
Oh yeah, sorry, no, you guys were discussing, so because this term of resetting and rebooting and reformatting and whatever, there has been some discussion about does cyclophosphamide doing most of the job already prior to the CAR T, and I'm not sure of any of the trials that I've seen so far that they are actually comparing the effect of this resetting, reconditioning.
Yeah, that's also a good point. Although we've been giving cyclophosphamide for many, many years, and we haven't seen anything comparable to what we see with CAR T cell therapy. So personally, I think that's very, very unlikely, because otherwise we would have seen this in the past. And we and we've been giving cyclophosphamide in higher dosages and as well, and much more frequently than just the preconditioning required for CAR T cell therapy. So although I think it's a good point, personally, I think it's very unlikely that the cyclophosphamide or the fludarabine are playing at least an important role they've obviously important, and that's essential that they are given, but it's probably for other reasons, and then I don't think they're responsible for that potential reset of the immune system. Otherwise, we'd have seen that in the past, and that's not the case.
That's great.
And some of the off-the-shelf products, there's no need for the ablative cyclophosphamide fludarabine regimen before. So that will be interesting to see if they will achieve this reset. And also the thought that some of these off-the-shelf products, they won't last forever. So unlike in hematological malignancies, you don't need to have such a prolonged B-cell depletion. We just need to reset. So that will be all interesting to see whether we can do this with least toxicity.
Great. I think Valerie also has her hand up, so I don't know if I want to give her the floor.
Hi, so thank you so much for organizing this. I was wondering, because we're talking a lot about what we fear and what might or might not happen, but I was wondering if some people in the room are actually part of a trial right now, and what has been their experience in setting up for, because at least for us, we're less excited because it's so hard to like get the infrastructure and the help and the support locally to really participate in any of these. So I was just wondering if people were actually doing trials in the room using CAR T cells.
I think that's a great segue, Valerie, to our next session. Yeah, I think the next session, I have this on my screen. I'm trying to remember CAR T like trial logistics. Go ahead, Leslie, if you want.
Oh, okay, sure thing. So our next session is looking at CAR T trial logistics as Valerie's wonderful segue into this next session. And so we welcome Mazen Damachki, who is going to be presenting a brief topical overview of the logistical elements of CAR T therapy. And then Dr. Gary Gilkison is going to present some lessons learned from interspecialty trial collaborations of mesenchymal stem cell transplant in lupus. So video, please. Thank you.
CAR T is a multi-step complex process in the way of getting the right participant identified, after which they have to undergo a series of steps, including leukapheresis, obtaining cells that are shipped for manufacturing, during which process T cells are enriched, then they are transfected using virus-based methods, and then they're expanded, after which they're shipped back to the site and they're stored until they're infused in the patient in a specified location. And some of these protocols require a conditioning event prior to the administration of the CAR T cells. We had negative experience with trying to address the logistics of a CAR T study, and we've learned from that experience. Initially, we were late in involving the cell processing and pheresis group, and it turns out they didn't have the bandwidth. Now we have been successful for the same study, which we failed initiating years ago, to initiate that same study later on as the bandwidth of the immune effector group and apheresis group got to be better. And we address several different logistics we didn't think about. A dedicated freezer, who will administer the cells, who will monitor patients after the infusion, where will everything take place? All of these are logistical questions. How long to monitor the patient afterwards? All of these have to do with local context, logistical questions that I, as a neurologist, neuromuscular specialist, need the help, the collaboration from the oncology team, from the immune effector cell group, as well as the pheresis group. So we were successful in initiating this study. And I think the next step is, well, what should we expect on the patient side from once we administer that therapy about the type of adverse reactions? One of the early adverse reactions that one can see in patients with cell therapy and CAR T therapy is the CRS, or cytokine release syndrome, which can be as mild as fever, but can be as severe as hypotension, leading up to respiratory insufficiency, requiring respiratory support, coagulopathy, and so on and so forth. These tend to happen in the first few days after infusion. And during that time, the IL-6 level is increased so much so that one of the standard of care is inhibition of the IL-6 receptor with tocilizumab. And for that and other monitoring and administration and management of this type of reaction, you can imagine where our folks from the oncology group are extremely instrumental. Now, the other side effect that is a bit more advanced is called ICANS. It stands for immune effector cell associated neurotoxicity syndrome. And this develops again in the early time frame, within a few days, up to 10 days. And it has to do with several different parameters that can influence the likelihood of it. It has a certain pathophysiology that culminates into diffuse cerebral edema, and it tends to follow the CRS. CRS develops first, then it can stop with the CRS or can evolve into an ICANS. And there's a variety and degree of severity, and in rare cases, ICANS can be fatal. So the lesson learned from our working with several CAR T studies in the last several years is that these are logistically complex studies. You have to have experienced immune effector cell therapy group that are involved early on. There's a certification process for centers to have that type of capability, but involve them early on. There's leukapheresis, there's shipping, there's receipt or manufactured cells. There's a GCP compliant storage that's required for that type of complex procedure. Also involve your oncology partners. There's a conditioning protocol. There's an infusion process that takes place in the hospital with oversight by the oncologist, as well as the neuromuscular neurologist or the rheumatologist or the physician overseeing the care. But you need to have that partnership with oncology taking the driver's seat. And in the hospital observation for managing the side effects and monitoring for side effects like CRS and ICANS is extremely important. Now, all of this can happen. It takes a lot of resources. It takes a lot of time, a lot of collaboration with oncology, cell therapy, apheresis, and with the hospital itself. Therefore, it's important to earmark in the budget the time and effort for the PI and the coordinator to really be able to effectively manage this complex magnitude of variables in these studies. But these are high reward studies, but high risk studies. When managed properly, you can do them quite safely at your site. Thank you.
Hello, my name is Gary Gilkison. I'm a rheumatologist at the Medical University of South Carolina in the United States. I'm the principal investigator of a trial, investigator-initiated trial of using mesenchymal stromal cells derived from umbilical cords to treat patients with refractory lupus. This trial is a double-blind placebo-controlled trial, which has never been done with MSCs before, and it involves 8 other centers throughout the United States. It is funded by the NIAID as well as the Lupus Foundation with some contributions by MUSC. I hold the IND for the trial, and of course, that was somewhat difficult part of it, and getting funding for it is also a difficult part, but those happen. But this conference is mainly about the CAR T cells, and so there's similarities in that they're both cellular therapies, which I agree is the dawn of a new era in treating different types of diseases. What they have in common is that they're both cellular therapies, both of which have to be neither are off the shelf, and so there's the challenge of logistics, getting the patient in at a particular time prior for screening, and then having the time to prepare the product before infusing the patient. That has gone well for us. We have our own clean cell facility here at MUSC that is preparing all the cells for all the centers, and then we ship them out to the centers overnight. So their logistics arranging that is a problem, but it's worked well for us thus far, and I'm sure it can be worked out with the CAR T cells. The advantage we have is that there's no need to prepare the patient with any types of chemotherapy. They don't have to, they're allogenic, you don't have to be matched in order to do it. The big thing is not needing any apheresis time. We are participating in at least one CAR T cell trial, and that turned out, or is the major roadblock for us entering patients, is the availability of pheresis to derive the patient's T cells. So we've been allocated the ability to have one or two patients in this. That can be overcome by some of the products that are apparently off the shelves or don't require the chemotherapy before conditioning. But overall, I think there's excitement about these things. The issue is the overwhelming number of companies that are entering into this market, with it already being over 40, and deciding which ones are going to be effective, which ones are having the least toxicity, as well as be economically feasible at this time. So, as been said by others in the lupus world, we don't have enough lupus patients for all these trials, and that is compounded by the difficulty in finding these patients that are fit for this type of aggressive therapy. For the MSCs, it's not as difficult because the patients don't have to be preconditioned. You don't have to pherese them, and MSCs have a proven track record of being extremely safe without side effects. So I think there's room for both MSCs as well as the CAR Ts. I think figuring out which CAR T approach is the best is going to be difficult. And the MSCs also, there are various groups out there pre-treating and doing other things to prime MSCs before infusion, and some of that may be better, it may not be better. But overall, I think you have to have buy-in from your institution, which we've been lucky enough to have here. We were lucky enough to have our own clean cell facility and a very good clinical research unit for which we can do trials in. So I think the main issues are, the coordination of things with the CAR Ts, and how we're going to pay for it, and how we're going to decide what patients get it. The other thing I haven't seen is whether there's going to be some type of a placebo arm to this, which will be very difficult to pherese a patient and precondition them, but then not treat them with the CAR T cells. I'm not sure how we're ever going to figure out in a controlled way whether or not the CAR Ts are totally effective or not. But I appreciate being asked to participate, and I'd be happy to answer any questions. My email is gilkison@musc.edu.
Thank you, Mazen and Gary. So at this junction, we can open the forum floor for community discussion. Please remember that there's the chat that you can put your responses, comments, new thoughts into. And so let's see, it's a little hard to see hands.
I think Karen wrote a nice kind of summary of what she had a chat with. Is Karen there? Oh, there, yeah. And Valerie. Yes, the challenges and the logistics that Valerie was mentioning it's very echoed by what Mazen was talking about. Oh, Karen, and you said like there was a huge sigh of relief.
Yeah, my rheumatologist was actually, he was like, oh, thank God. Bless him. He would have done it for me anyway, but
yeah. Oh my God, that's hilarious. Valerie, I don't know, I want to put you on the spot, but like, you were talking about in your institution that it's been really challenging, and maybe that'll open up.
Yeah, I mean, even for just clinical trials, it's a challenge, even if it's just infusion of drugs. So for us, it's, but then the notion that people need to stay in the house like for a couple of days, for us, it's just, I don't think it's ever going to happen. But I know that in Canada, there's some of our colleagues in Ontario that are trying to also create a network of not just rheumatologists, but hematologists and that are using CAR T cell for oncology, but try to put all of these people together as a network to create more knowledge and maybe try to decide which type of trial and which approaches as a group we want to focus on, and not try to do everything everywhere all at once, and like have different centers doing one thing, but try to coordinate as a group interested in CAR T cells. So maybe we get experience, or at least try not to get all over the place and then not get any answer because it's too few cases done. So I think maybe it's something, it would be a way of trying to I don't know, focus our efforts and get more answers. But yeah.
I think Pedro had his hand up next.
Yeah, so just to echo a little bit what Mazen said, I think we don't have the expertise. We rheumatologists, neurologists, dermatologists, pediatricians, we do not have the expertise, full stop. So it's essential to find a shared care model with those that have the expertise. And those are the cell therapies team, the hematology/oncologist, depending on your specific setting. And when I was initially approached regarding these trials, my immediate thought was, no, that's impossible. I do not know how to deal with this type of medication. What made it possible was the engagement of the cell therapies team, the hematologists. And I was lucky enough to have a team here at UCLH that have treated a lot of patients with hematological malignancies with CAR T cell therapy and other cell therapies, and they're doing so in many clinical trials. And due to that engagement, that the shared care model that we agreed, we're now able to do them. We just treated 4 patients with lupus recently with CAR T cell therapy. We have 2 myositis CAR T cell therapy trials that are probably going to start early next year, and then maybe a couple more in lupus. But the only reason why I'm able to do it is because of the engagement of the cell therapies team, and they've got the setting, they've got the expertise, and we've agreed a shared care model to do it, and that's fundamental. I think that's essential.
It's so important, and it sets expectations, so that may be the best way forward. Gary, you had your hand up next, and then Chet.
Yes, I echo what was just said. The issue that we often have, or I'm not sure how often it is in the US, but our people that are doing the CAR Ts are overwhelmed with cancer patients and others, and don't have the bed space to do it and don't have the time to do it. Although they've agreed, like I said, to do 1 or 2, and the pheresis was the other thing. And the other, we were talking about B cell, resetting the B cells. And I think that
is partially true, but I think we aren't far enough out to know whether the B cells are going to make the same mistake they did when the patient initially developed lupus, which harkens back to these treatments with bone marrow transplants in lupus, where they were treated and ablated and then received the transplant. And in that instance, they did well if they survived the procedure, but then most of them do recur, maybe a little less severe. But I don't think, if that's the model that we're going to see that that's the case, that they're going to be permanently reset, but that they'll be temporarily reset. And we have people, Naki-sans and others that are the B cell efforts we're working with to really look at that, to see, how the B cells are recovering.
Thank you. Chet.
Can you hear me okay?
Yes, we can, Chet.
Okay, so I don't want to be too redundant here, but it's just when you shoot at a target that's still, if you have an arrow and you shoot it, that's easier. When you shoot at a target that's moving, it really becomes difficult. And this is a moving target. And I guess I would emphasize logistics of something like this. And I do think that the problem here is not only, it is logistics, and I think this is only going to be able to be done at very select centers. And the problem is that pharma, throws the net out in a very broad fashion, as they always do. And think about it. When we are involved in trials, we oftentimes are involved with one company that has a particular target. This is a gorilla that has many different companies that are going after different centers. And as I said, I really think that there's going to be only select centers. And pharma, they're going. We've never had anything like this. Now, think back 20 years and all the things we have available to us today in terms of the targets and the different companies. This is even different than that. Many different companies are going after sites, and I do not believe that many of the sites that they're going after are able to do this. And I just fear, my concern is that there are so many that will maybe sign up and they won't be ready. And the logistics will be outrageous. Just think about the logistics of location. Maybe even the oncology isn't really in the same location as the autoimmune group. I mean, that's just a simple thing in Pittsburgh that we're dealing with. So, this is there are, as often happens in a forum like this, more questions are raised than there are decisions and things that are able to be resolved. So this is going to be very few patients, and I hope that it's going to be very few centers, because this can get ugly really quickly.
Chet, thank you for that. And I will just echo what was quite a repetitive input on those surveys, which was standardization of safety, standardization of quality. Never mind these nuts and bolts, brick and mortar logistics and financial logistics. It's how are we monitoring safety implementation and guarding efficacy through standardization of implementation.
And I didn't even mention, I'm sorry, just add one other point, didn't even mention budgeting. And if you don't have somebody that's paying attention to budgeting for this, you're going to get stuck, because, you got coordinators that, we don't have the coordinators really necessary in autoimmunity to be able to handle this. The coordinators really have to come from oncology, and they have to be ready and primed to be able to do this, and that is another major logistical issue.
Well, thank you, everybody. for such really rich conversation. I think we can now move to the next session.
Thank you, Leslie. And so I will be sharing this session with Ingrid. Are you there, Ingrid? Yeah.
Yes, I am
okay.
here.
Okay, so in any case, we will start with our videos and with patient perspectives. And so there are two videos. One will be shared between Marianne de Visser and Leslie, and the second one will be shared between Ingrid de Groot and Karen Schenk and Angel. So if you could please. Yes, thank you very much.
Leslie and I will start a short presentation in which a number of important aspects of CAR T-cell trials will be discussed. Next, please. This slide shows a paper, a letter to the editor actually, which claims to be the first publication on a case of myositis as part of the anti-synthetase syndrome undergoing CAR T-cell therapy. There are other cases, of course, however, most of them were reported by the same consortium. The patient, a lady, had a 6-year history of muscle weakness, arthritis, skin changes, and ILD, for which she was treated with a host of medications, as you can see on the graph at the bottom on the left. Continuous corticosteroids, and in addition, various immunosuppressants, B-cell depleting therapies, and courses of IVIG were subsequently administered. And there seemed to be short biochemical remissions if we look at the CK. However, she was considered refractory, and therefore CAR T-cell therapy was tried. And this led to a severe B-cell depletion, to a steep decline of CK, a marked improvement of the patient global activity and also of the patient global activity, albeit to a lesser extent. Major treatment improvement was achieved, and you can also see that the MRI improved markedly. Follow-up in this patient was 150 days.
So this and other cases come across to us as being quite remarkable to have a remission without other treatments. One of the questions that this case brings up is from those of us from our very different geographical backgrounds and practice centers' practice habits, is that our practices are very variable over time. And for example, in our center, we can see in this graph that there were a single treatment administered after single treatment option. Whereby in our center, for example, our practice is different, whereby we might begin with 2 or 3 treatments, and we try to glean and predict the severity and the rate of progression that this presentation might be suggestive. So in this case here, what we're seeing is a patient that's described as refractory, but in fact, we really don't have a definition as a community for refractory. And how do we assess multiple treatments over time, the need for multiple treatments over the time, and what parameters are we using for treatment switching or treatment adding? And then there's the case where I think everybody reading this case gets the feeling that this case is this patient has a very progressive, active disease. But in other cases, it may not be so clear. And what is our criteria for disease activity compared to a patient that may have had delayed treatment, therefore allowing a significant amount of damage to accrue, which is not reversible, but leaves behind tissue damage and also leaves behind significant symptoms. And the other question that this brings to mind is, how do we select patients for progression? So we also saw here in this really exciting case that there was a remission of about 120 days, 150 days. And but what is our definition as a community for remission or suppression of disease?
On this slide, you can see some other reports, also focusing on another subtype of myositis, immune-mediated necrotizing myopathy, one from the previously mentioned consortium, and one from the group which has initiated the RESET myositis study. And apart from CD19 targeting, there are also some reports emerging on BCMA targeting cell therapy. So if we consider what the implications are of CAR T-cell therapy for people living with myositis diseases, then we see that there are a few case reports and small case series on patients with myositis in the context of anti-synthetase syndrome, or as mentioned, immune-mediated necrotizing myopathy, which show that CAR T-cell therapy may hold promise. But as mentioned by Leslie, follow-up is so far relatively short. And in addition, CAR T-cell therapy can be considered a high-risk treatment, which has to be performed in hospital.
Yeah, thank you, Mariana. There's maybe serious yet unknown complications. And, if we're looking at the connective tissue disease population compared to the oncological population, we have some really strong lessons learned from connective tissue disease in hematopoietic stem cell transplant. And so do we need to be looking closely at these lessons and considering them for examination when using CAR therapies in connective tissue diseases? And I'm speaking most pointedly about cardiopulmonary involvement that could impact the heart and the heart's ability if the patient during a conditioning regimen were to get a septic infection, how well are they equipped to handle fluid resuscitation, which is what we saw in the scleroderma population, that occult heart involvement from scleroderma and diastolic disease and other factors. The fluid resuscitation itself brought out a lower performing ability of the heart to manage fluids, and this is where we saw signals elevating in death related to fluid resuscitation with ventricular arrhythmias emerging. So we have to ask ourselves in these patients, do we need to be considering cardiovascular involvement? Do we need to be considering how much of their lung disease is damaged, and is that damage predisposing them, one, to higher infection risk, two, cardiac function risks? Thank you, Mariana.
Another very important aspect is how do we select the appropriate patients, and what criteria determine their eligibility. So lots of research has to be done first to determine which patients are really good candidates for this therapy. And previously, Leslie already mentioned that we do not have an appropriate definition of when we consider patients refractory. And that is something we have to achieve consensus on before we start these high-risk treatments.
Thank you. So I guess as Mariana is leading towards, are we moving a little bit too fast with the development of these clinical trials to ensure the safety of the largest amount of patients? And do we need to be accruing more data on a smaller amount of patients related to safety, related to implementation of the procedures, related to development of a subsequent second autoimmunity as a consequence of this, resetting, as it were, of the bone marrow? And then the other critical questions are related to patient vulnerability. Some of our populations are more vulnerable than others in the face of treatment and care. And there has been discussed in this session pressures by sponsors to recruit. And that makes newer investigators feel vulnerable to supplying what's being asked of them. And that could result in a downstream pressure that on the patient, whereby perhaps a patient that is really not the best selection for a CAR T trial is enrolled, or their particular presentation might be better treated with lesser aggressive therapy than CAR T. So these are important questions that have come up in our discussions together. So how do we make sure that patients truly understand what we're saying to them? And there's increasing data now that demonstrates that, you know, even on the ACR website, even on the American Medical Association website, we don't do a good job. And we hand patients, you know, very thick bundles, which is called a consent, which is a lot to handle. The centers, the NIH has recommendations for readability, and this is a health literacy manual from the American Medical Association, but a recent study in November revealed that evaluating all the patient education materials on the AMA website and on the ACR website did not meet the benchmarks of the American Medical Association. So the American Medical Association did not even meet their own benchmark of a sixth grade level, which I think is about 12 years old, just so our non-US colleagues can understand what that means. And this study showed that there was nary a patient education material that was less than 8th grade level. And then prior to this was also demonstrated in 2021 as something very similar, a 20-year analysis that demonstrated that only 2% of all these studies that were geared towards these educational materials that were geared towards patients only 2% met AMA standards. And then finally, this last study in 2019 looked at rheumatological patient education materials in the UK, Australia, and Canada. And then they tested patients afterwards. They asked patients their perceptions and they tested them afterwards. And the assessments were dismal at or around 50% for these medications. And these were sheets that were related to biologics, NSAIDs, prednisone use, methotrexate. And the patients overall stated that these sheets, each of them were very difficult to comprehend.
So Karen, what I'm most concerned about with this CAR T-cell therapy is the lack of lay information for our patients. The average patients, they see all these headlines and all these promising articles, and they're not in a situation where they have the skills to evaluate that information. So they only see what they want to see, basically, and which is hope, or what the other party wants us to see. And at the same time, what we need, if we really want to talk on an equal basis about this kind of thing, is that we need some kind of information. So do you experience that too? Do you hear that from other patients as well, that there's this lack of information? What exactly are we talking about? What is CAR T-cell? How does it work? Are there adverse events? What are the long-term effects? What types of myositis would benefit? That sort of thing.
I think that what I have observed is very variable information as well. And I do agree with you that a lot of the headlines are very impactful or pro-CAR T treatment, because this is also coming from the experience of people who have seen the successes in the oncology space. And for sure, there are a lot more years of experience. There are a lot more patients who have benefited, and also many patients who have unfortunately passed away in CAR T-cell treatments in oncology diseases. So we are in a place now where a lot of the positive messages that we hear and we see on media, in social media, it's coming from another type of disease. And I'm not sure that the patients are fairly informed because this is a very complex disease treatment, a type of treatment. It's not the kind of treatment that we are used to seeing in our disease area, which might be daily pills. It could be even weekly injections, monthly infusions. This is not something that we as an autoimmune disease is used to seeing. And I think that there are definitely lessons that we can learn from the cancer community, but to misinform patients in our disease area, I don't believe that is currently fair. A lot of hope is there as well, as you have mentioned, Ingrid. And the patients that we know who are the sickest or the most desperate for a treatment that actually works because they have spent years suffering from all sorts of symptoms. You know these patients very well. You and I have both been these patients.
Yeah.
And I'm sure that if we were at the worst of our disease, we might feel more willing to do a very special kind of treatment like this.
Yeah, but that's also the bit that worries me, because you and I, we're above average, I think, informed about all these things, because we're a patient research partner. You even had medical training. I didn't, but at least because of all the things I do in my myositis community, I have, I think, a bit more knowledge than the average patient. And still, I do not fully comprehend the impactfulness of this therapy. I mean, and what I hear a lot of other patients, they pick up that it's been used in cancer before, sometimes successfully. And that's what they cling to because, as you said, they're vulnerable. But at the same time, they also think, yeah, but myositis is not cancer, is it? I mean, that's also a bit confusing. And as you say, this treatment is an option when you're at the worst situation possible with your myositis, which makes it an even more vulnerable situation.
I agree. I think when they think of cancer patients, in a way, maybe autoimmune diseases does not sound, as fatal as a cancer. And maybe this could also be misunderstood, that I don't have cancer. I only have an autoimmune disease. My chances of surviving this could be probably better. But yet, like you said, not every patient really understands the pros and the cons, how bad things could get, how the immune system reacts to a treatment such as this. I agree with you that lay language materials need to be created to really explain to patients. I do hope that the companies and the institutions that are planning to do these trials are providing the patients either in written form or in videos, or even just sitting down and explaining it properly in lay language, that they can have the time, to ask the questions and to really understand what they're getting themselves into. My fear is that this is not really the case. I don't have any direct evidence of this right now, but I do feel that we need to be more supportive of our fellow patients, and also to help them understand that this is an option for them. They must ask the questions that they want to ask. And if they don't feel well explained to, they shouldn't, put themselves at a risk for something that they don't fully understand. Yeah.
And especially since, I mean, it's rather new. So there's not much they can say right now on the long-term effects, because most patients have only recently been treated like this. So that's another thing. But like you said, there's a huge unmet need for lay information, maybe even animations or whiteboard videos for people who are not so literate, or and make sure that other language can profit too. Yeah, but I agree with you there. There's also a task for us as patient advocates and patient advocacy organizations and institutions like MIHRA to be ready to do this, to inform the patients and the public in general, the loved ones of the patients who have to deal with this as well. So, yeah.
Yeah, I personally have a knowledge gap. Like you said, despite the fact that I do have some medical training and I do understand more than the average patient on the scientific processes involved in such a treatment, but I do admit I have no idea what happens after I get admitted to the hospital and I do have what they call the preconditioning treatment, and they take my cells and they take my cells away and they do something to my cells and then put it back inside me. That process for me is not clear. So I'm 2 weeks maybe waiting at the hospital, nearly living in a bubble because my immune system has been completely depleted. So I'm also very susceptible to infections. I have to take a lot of care 2 weeks is a long time to be in a hospital. As you know, hospitals are little factories of bugs.
Yeah, true. Yeah.
And during that time, I will be in the hospital, hopefully very protected. But then my cells are somewhere.
Yeah.
It could be nearby, it could be even in a different part of the country, and they're being tweaked. And they're going to be put back into me with the expectation that they would multiply and, you know, come back in a much altered state to fix the problem that, to be honest, nobody really understands what is the problem and what causes myositis. So my fear is that if we don't fully understand the process, you know, we are signing off blindly. We're signing off to something that we don't actually know. And it's okay if physicians and medical teams tell us that actually we don't know ourselves. I spoke with a physician recently and I said, hey, you approach me for this study, but do you know what happens once they take my cells?
Yeah.
And he said, to be very honest, I have not visited where they do this.
Okay.
I don't have a good visibility of, you know, I trust that the company will take it and do what they need to do it, you know, with it and give us back a good quality product that we can put back into you.
Yeah.
A lot of the decisions that we make as patients, we do it in good faith and good hope because we trust our physicians and we trust the medical team that we work with.
Yeah.
But these are the gaps that I think is still missing.
Yeah. And you mentioned two important things there, like one of the pre-treatment, as you called it, the isolation in the bubble. Now, I know from a personal experience with COVID, I've been isolated for 11 days. It's very impactful. So that's one thing to consider. And the other one, yeah, what happens to your cells? That was nice and very honest of your physician to tell him he didn't actually know. But you also said something about trust, which triggers another thing in me, because we're talking shared decision-making. That's what we're after most of the time, which means there's no power imbalance, there's equality. And to do this, to make a proper decision or shared decision-making, to do that properly, you need to take away the power imbalance. At this time, I think there is a huge power imbalance. And as long as it's there, I don't think you can rightfully say that CAR T-cell can be a shared decision-making thing, or it's very, at least very challenging to say so. Do you agree, or is that too bold?
No, I fully agree, but also at the same time, I think the expectation to completely be on the same level is very challenging. Every patient has a very different background and a very different history, and I think institutions which have been successful to date with the handful of patients in myositis, they currently have that supremacy, as you put it, because they can say that they have experience and they know that their patient is responding. And yes, it's a good observation for sure. My feeling is just that these successes, despite how few they may have been, have driven this very fast uptake by many industry players, private institutions, smaller, cell companies, because they do see it as an opportunity to create a new revenue stream.
Yeah, that's another thing you say there, Karen, that things are moving fast. They're moving like really fast. And I'm not sure if that is all just a good thing, because there is so much competition. That's not always good, not for patients at least. Because one thing I'm wondering about is, are all these studies, are they comparable? Which outcomes do they use? Did they even involve patients in trial design? And I don't mean tokenism here, that they had design already and they wanted to have your signature and said, oh, I've read it, I'm okay with it, but really thought about it. Which outcomes matter to patients most, and how feasible is this study and the intervention? And
yeah, Ingrid, I can only say this from a perspective of, I hope they have, because we've seen now, I think the last time I looked at clinicaltrials.gov, there are about 14 or 15 CAR T trials, and they're international. I mean, they're posted international. There might even be others that are not yet on clinical trials that are being discussed and talked about. I personally have not been reached out to help in the pre-protocol design. I don't know if you have maybe, or maybe some of our colleagues, worldwide in our myositis patient organizations worldwide, maybe they have been approached. But I think this is where most recently at a conference in the US, where there was a session called "Did we put the cart before the horse?" You know, and I think in many cases, trials are already, the conceptualization of a trial has been done in the scientific community, has been done with the industry partner. And then patients, like you said, the word token, in many cases, we do come in quite late in the process. And I think this has to be changed. I think we must be able to, you know, voice what is important to us at the start before a pen has been taken out to really start to design a trial. I think it's starting to change. I do see the positive changes that are implemented now, that more and more patients are being invited to trial steering committees or patient panels in companies. I think this is a good change. But again, before such a different trial takes place, I would have hoped that many of us have been contacted before the trial is actually public and before the trial has already had, you know, investigator sites set up, because then there's no going back now. Or maybe even if you wanted to go back at this point, it's going to be very difficult.
True. But maybe there's a nice task for the MIHRA community there.
Absolutely. I think there's definitely more we can do in MIHRA with also many other groups that are working in myositis. We really should come together to solve this problem.
Yeah.
That things are running faster than any of us can keep up with at this time.
And still keep our hopes up, because it is potentially a very promising therapy. So we mustn't forget that. Yeah.
Okay, so that was fantastic testimonies from both the patients and also colleagues, and you know, this is now open for discussion. Maybe I'll start with the comment. I was listening to Ingrid and Karen about how difficult it is for patients to understand these concepts and what is really CAR T cell therapy, what does it involve? And to be honest, those comments apply to rheumatologists and doctors in general, because this is also new for them. You know, that's the reality. If you ask most of the busy rheumatologists doing general rheumatology clinics to explain to you what CAR T cell therapy is, what the chimeric antigen receptor is, how it is engineered, they don't know, you know, because this is also new for them. So then I think that's the part of the problem, and this is all new for patients, but also for rheumatology, yes, and neurologists and dermatologists, yeah, doctors in general. Although, you know, that's the reality, and you know, I recently, as one of the chief investigators for one of the CAR T cell therapy trials, I participated in an ethics committee meeting, and I think 70 to 80% of the meeting was about communicating this to patients. So they were spot on. They were spot on. It was about the patient information sheet. It was too complicated. We needed to simplify it. It was asking me how was the consent process going to take place. So they were spot on, and it's difficult. So one thing that I'm going to do is to the consent process will be shared with the hematology team. So it will not be only me, it will be us. And I think that's really important, because they do have all the expertise in oncology, and I think it's important that they also share that expertise with the patient. So that's one of the things that we're going to do, and we're also revising some of the patient information sheets to make it a bit more simple. But it's not an easy task. It's not an easy task, I have to say. It's really difficult, and I think an important part of it will be the actual face-to-face interaction, and making sure that it's a multidisciplinary face-to-face interaction together with the hematologists.
Thank you, Pedro, for that. Is there any raised hands? I don't think I can see any.
Yeah, me neither.
No. There is a lot of chatting going on, which is a good thing in this case. But that's also another, it was on shared decision making, but also in other things that have been discussed in sessions before. So I'm not sure if we need to dive into these, unless someone who put something in the chat want to address us all and emphasize his or her concern or remark.
Oh, Leslie, got a hand from Leslie.
I guess I would just clarify my concern is that, well, you know, shared decision making is something that demands time, and it demands its own time, and it is going to be different for every patient. And if we're truly going to follow some of the advice that has come up in the surveys from patients and supporters, we have to be prepared to protect that time in order to be able to truly listen to somebody's concerns. And unfortunately, you know, I don't know how it is in different places around the world, but there's always somebody standing on your neck because you're not moving fast enough. Regardless of how complex our patients are. So fighting for that time to be able to speak about, number one, the complexity of the disease alone takes time. But then when we're getting into complicated therapies like rituximab, IVIG, and now we're talking about something very extravagantly complicated. I guess my point is that we have to think about that time needs to be reimbursed, because it's going to be very difficult for investigators to fight for that time from their hospital administrator and from their divisions, as it were. Because they're paying for our time, so it has to be somehow reimbursed. So that's a budgetary consideration in order to give patients what they need. That was my comment in the box.
Thank you for that, Leslie.
We're waiting to hear for more comments. One thing that I have in the way of idea, surgeons, when they do complex surgical procedures, they have a pre-recorded video for the patients to watch and then have more detailed insight about what this is going to entail. I wonder if from the patient's perspective, again, I'm not on the patient side, it would be helpful to have some kind of a video that is looked at even before they come for consenting, to just know what they're getting themselves into, to have an idea about the complexity, all the moving pieces and the processes that are generally for the study, but also specific to the site. I wonder if that's not going to be something that would be helpful to clarify some of the complexities. And I have to admit, you know, sometimes, you know, you see a patient, you think they're a good fit on paper, but when you evaluate them in person, it's just not the right person for the study at all.
I think that's also highlighting wasn't like the key, like when you're on these ad boards, I'm surprised. I think some companies are really good about making sure there's a patient partner there, but on some of the ones that I've been on for NK or CAR T, there's not even a patient, you know, advocate there or a patient. And I always tell them to reach out to the TMA or the MSU, or anyone else, because this is when they would tell us, like, they're just concerned about recruitment and how are we going to get in and get this set up. But if you don't have any patients who are going to want to do this, then you're not going to be able to finish your study. So I think it's also on the onus of us to remind these companies who are so hungry to get these trials done, to bring the patient back into those key advisory meetings as well. I don't know what people think about that, but yeah.
And at an early stage too, I think. Yeah. To avoid tokenism. Yeah. Any comments on this or?
Leslie has a hand raised. Leslie?
Oh, okay. I'm going to yield Chip, and I'll speak after Chip if there's time. Chip had his hand up after me, but he hasn't spoken yet.
Yeah, and one of the things I would like to say from a patient perspective, I have IBM and I go to Johns Hopkins. And from a patient perspective, a lot of the information we get on the trials, the information we get on what we need comes from our rheumatologist, comes from our team that we get to meet with, whether we're going to a conference, whether we're listening to a podcast. A lot of the information, that's who we come from. We build trust in the providers that we get to see. When I travel to Hopkins, I trust the ones that I get to talk to guide me and tell me about the trials that are coming up and let me know the pros and cons, because they know how IBM has affected me individually. So from a patient standpoint, it's the knowledge that is transferred to me by the scientists in the community, and they say, yes, this would be good for you with the stage that you're going through, with what you're experiencing and where you are in the disease, rather than me just going on feeling and hope and say, hey, CAR T sounds great to me, let me go see if I can try it. We have to build that trust, and we have to have that conveyed to us as a patient.
Thank you, Chip. Thank you. Leslie?
I guess that all comes back to the quality of the information that investigators are getting, the quality of the information that's being handed to patients. I loved Mazen's idea. We did something like that with sarcoidosis, one of the sarcoidosis trials with a video. But the issue is, and having, like I said, you know, I get involved in some of these patient education material development, and you know, there's the input from the beginning is so important, you know, and it's a paradigm throughout our clinical investigations, guideline developments, that there's just still, the patient voice is still tokenized. It's divided at the last minute. There's guidelines presented to them as a fait accompli, gathered from limited data of systemic literature reviews, and then investigators, experts saying their opinion, and then we hand them a slate and say, do you agree? Do you not agree? So, you know, it's, and this is just the same with trial design. We would definitely, in my opinion, be developing more feasible trial designs, having higher patient retention if we had patients from the get-go, as Karen Chang has said, before you pick up the pen to write the design. And that's where we need, so, and I think, I strongly feel, having been involved with industry, you know, we can't help ourselves, we're human, we have bias, we have agendas. So we have to support our industry partners and take that burden away from them and work as investigators, as experts, investigators and patients together from the ground up, developing these educational materials and then vetted by pharma.
Thank you, Leslie. And maybe there's also, like we said in our video, there's maybe a role that we should take up as patients as well, as patient advocacy organizations and as MIHRA. I don't see any more. Nope. So I think for this session, all has been said and shared. And I'm not sure, I think we are over the 2 hours already, but not sure how to proceed from here.
So I think, yeah, we're now at the end of the planned session. We do have, so feel free to leave at this point in time. But for those that still have any comments or would like to discuss, would be happy to stay a few more minutes. But unless if nobody else has a comment or question, I think it'd be time to close the forum. Right, Leslie? Do you agree? Yeah. Okay, so Ingrid, would you like to close it? Shall I give you the honor? Or shall I
Thank you,
do it?
Pedro. No,
Yeah. Go ahead.
I just want to say thank you, everyone, for first of all for including us and the patient community, which is very much appreciated. Thank you, everyone, for being here on your maybe even your sparsely free days in the weekend. Thank you for attending this session and sharing your thoughts, your concerns, to be honest, with us. And Leslie or Angel, I think you should be the ones to officially close this. So the floor is yours. And thank you, Pedro.
Thank you, Ingrid. Final word, Leslie.
I am moved. As an investigator, as a community member, I am moved by our voices. And again, we do better together, and we do better together and moving forward and hearing each other. And this has been really fabulous. Thank you for this really rich time. And thank you to Angel Soto for being our wonderful production engineer. And this will be posted on our website and maybe on a YouTube channel, so it could be shared with patients and maybe get more information, as well as the surveys will still remain open, and we'll be coming back to you with next steps. Thank you.
Excellent. Have a good weekend, everybody. And see you soon.
Bye, everyone.
Bye.
Bye.